All right, so now we're going to get into Chapter 6, which are acellular pathogens. So remember, acellular, A means without, and cellular is a cell. So things that are without a cell. So basically what we're going to be talking about here mostly are viruses. So we're going to get to 6.1 here. So this first portion in 6.1 is kind of giving us the background on viruses. So they're very, very small. Viruses are much, much smaller than bacteria and other prokaryotic organisms, and of course eukaryotic organisms. So they were not able to be seen with light microscopes until there was a filterable component that was smaller than a bacterium, and that was first seen with the tobacco mosaic disease back in 1892. So at that time, this gentleman here, Dmitry Ivanovsky, who's a Russian botanist, he discovered this TMD, this tobacco mosaic disease in the plants, and he came up with this porcelain filtering device. This is called a porcelain Chamberland filter. And he made this porcelain Chamberlain filter. It had a pore size of 0.1 micrometers. So this allowed it to get rid of any bacteria that was greater than or equal to 0.2 micrometers from anything that was passed through it. So he took materials from this tobacco plant, kind of ground it up, mushed it up, and filtered it through this device. And what it did was it removed all bacteria that were greater than this size. And then what they found, though, that fluid that came through it still had the disease. It still infected other plants. So it was surprising to everyone that when he did this, that it didn't take out the disease, because they thought it was a bacteria, and they knew that it would get rid of the bacteria. So then over the course of time, what they did was they figured out it was obviously smaller than that. They thought that maybe it was some sort of toxin, and then they actually named it Latin for poison or toxin, which is a virus. So eventually it was determined that it was a virus. Today we can see that using electron microscopes. But this gentleman, Ivanovsky, then is credited as the original discoverer of viruses. So basically the founder of virology. Remember, that's the field of studying viruses. And so you should know that Ivanovsky, because he came up with this porcelain filter that filtered out bacteria, actually found or identified the first bacteria. Even though he didn't actually see it, he's the one that came up with the mechanism and actually figured out that it was something much smaller than a bacteria. Today we use electron microscopes to see viruses. We know a lot more about them. Viruses are distinct biological entities. However, we still don't know their origin. We don't know their background. We are not able to do taxonomy with them. We don't know their background, so we don't have a tree of life, not only because we don't know their background, but also because they are not life. Because there is no tree of life, because they are acellular. And remember, having a cell or consisting of cells is part of being alive. So viruses are not classified as being living. So since they do not have cells and they are not living on their own, in order to survive and reproduce, viruses must infect a cellular host. That makes them an obligate intracellular parasite. So obligate meaning it has to, it's obligated, and then intra meaning within, and then cellular, intracellular. So in order for a virus to exist, it has to be within a cell in order to be a parasite, in order for it to continue living and reproducing. So the genome of a virus then is going to enter the host cell, and then what it does is it directs the production of viral components, proteins, and nucleic acids needed to make a new virus. So it gets into the cell, it takes over the cell machinery, so all of the things that we know about a regular animal cell, human cell, plant cell, bacteria cell, how they have the nucleus, and then they have the different organelles like the Golgi apparatus and our endoplasmic reticulum. All of those different things, it takes over those things. The genome of the virus gets into the cell, takes over all of the components, or takes over all the organelles, and then directs the production of these organelles to make its own components, its own proteins, and its own nucleic acids. And then what that does is it makes these new viruses that are within this cell, and they're called virions. So new virions are made in the host cell by assembly of these viral components. So the virus gets in there, takes over the machinery of the cell, makes it make its own parts and then assembles those parts. And then these new virions transport to the viral genome to another host cell to carry out another round of infection. So then it just does it again. Once they're built, they've been assembled, they burst out of the cell, and then all of those new viruses are going to go and infect all new cells and do the same thing over again. So here are some characteristics of viruses. We're going to go through these, but first they are infectious and they are acellular pathogens, so we already talked about that part. We also just mentioned that they are obligate intracellular parasites, so with host and cell type specificity, and we'll talk about that in just a moment, a little bit more detail. They have DNA or RNA genomes, but never both. Their genome is surrounded by a protein capsid and, in some cases, a phospholipid membrane studded with viral glycoproteins. We're going to look in more detail at that. And then we'll look in more detail at these lack of genes for many products needed to reproduce, and that means that it has to be in a host cell in order to reproduce. So this is kind of the general overview chart of viruses, some characteristics, and we're going to go into the details of those as well. So first let's talk about how it is that we get a virus. So how we have a host, because the virus can't live on its own, and then how that's transmitted. So viruses can infect every type of host cell, so including plants, animals, fungi, protists, bacteria, and archaea. So we see viruses across the board basically infecting cells. Most viruses, however, can only infect cells of one or a few species of organisms. So a virus that can infect a protist, for example, likely will not be able to infect an archaea or a plant and an animal or bacteria. So usually it sticks to one type of species. And actually, what we see mostly is it only infects very specific hosts and only very specific cell types within that host. So, for example, a human virus may only very specifically infect a certain type of cell in the body, a certain type of tissue, rather than even infecting other tissues within the body. The range that an organism, or that not an organism, that the virus can infect is called the host range. So obviously range meaning the kind of arrangement, the number of hosts that a virus can infect is called the host range. And again, that usually is only one host or even a specific cell within one host, but some can do multiple. The viruses that infect bacteria are called bacteriophages, or simply just phages. The word phage comes from the Greek word to devour. So they go in there and they just take over, devour the whole bacteria, and then it bursts, and then they have more viruses infecting other bacteria. Other viruses are just identified by their host group. So, for example, an animal virus or a plant virus or a protist virus. Once a cell is infected, the effects of the virus can vary depending on the type of virus. So it really, really varies, and we're going to look at different types of viruses and see what they do to their host. But generally, virus can cause abnormal growth of the cell. So when it takes over and it gets in there, the cell can grow abnormally. And it can also cause cell death. Most often we see cell death. The virus gets in there and it's going to take over, make itself, all of its different parts, put them together, and then burst out of the cell. And then that bursting out of the cell is what generally causes cell death. However, in the process of it doing all of that within the cell, we can see this abnormal growth of the cell. We can see an alteration of the cell's genome, which can lead to things like cancer and things like that. Or it can cause little noticeable effect in the cell. So viruses can be transmitted through direct contact. So if one person has a virus, and say they just coughed into their hand, for example, and then shook your hand, then that would be direct contact. So directly touching something that has the virus in it can then get the virus in the next host. Indirect contact with fomites. Fomites are just objects that are likely to carry things like a disease or like a virus, like somebody else's fork, for example. So somebody that has a virus was eating with a fork, and then you went and picked up their fork and used that fork and got the virus in yourself, then that would be indirect contact. or through a vector. So the three ways that we can transmit is through direct contact, directly interacting with the virus, indirect contact, meaning picking up something that has the virus on it, or through a vector. And a vector can be an animal that is going to transmit the pathogen from one host to another. And then the vector portion, the vector type of transmission, can be done in two ways. We can either have a mechanical vector or a biological vector. So these vectors that we see are typically arthropods, kind of like mosquitoes that can bite us and then get that into our body, ticks, and flies. Those are some of the typical vectors because these things are going to be flying from person to person, biting them, getting blood from them, for example, and then biting the next person and then transmitting that virus from biting one person that has the virus and then biting another person and sending that virus along. So the two different ways that you can transmit a virus via vector, the first way, mechanical vector. So mechanical transmission occurs when that arthropod, so that animal, insect often, carries a viral pathogen on the outside of its body and transmits it to the new host through physical contact. So basically, for example, think of a fly. If a fly lands on somebody else and has that on them, has cell stuff on them, picks up a virus and it's attached to its legs, for example, and then lands on your food or lands on your eye or mouth, you accidentally get it in your mouth, that kind of thing, then it can transmit it that way. So it's mechanical. It doesn't have anything to do with the biology of an organism. It's just basically it's on something and you get it because it was on it. But it's through a vector rather than through fomites, which would be like a fork or something. Then there are biological vectors. So biological transmission occurs when the arthropod carries the viral pathogen inside of its body and transmits it to the new host through biting. So this is a good example of that would be the mosquito. So if somebody has a virus that's floating around in their bloodstream and a mosquito bites them and sucks up their blood, they get the virus inside of them. And then when the mosquito goes to another person that doesn't have that virus already, sticks their proboscis in the body, and then starts to take their blood up, there can be some transmission of blood in that way, and then transmission of the virus. In humans, a wide variety of viruses are capable of causing various infections and diseases. So we see a lot of infections and diseases with them. Some of our deadliest emerging pathogens in humans are viruses, so they're very difficult for us. We have very few treatments or drugs to deal with viral infections, so it makes it really difficult for us to get rid of viral infections and stop them from spreading and infecting others. Viruses that can be transmitted from an animal host to a human host cause zoonoses. So a zoonoses is when you have a virus that infects an animal. So for example, a deer out in the woods, it infects deer. It generally stays in deer, a virus for deer. And then somehow that is going to be transmitted to humans and then is able to infect a human. For example, we saw this avian influenza virus, the avian flu or the bird flu. It originated in birds, but then it went through zoonosis, or it is a zoonosis, and it can cause disease in humans. So it transferred over. It somehow made itself able to infect human cells rather than just bird cells. Then we also see reverse zoonoses, and that's caused by an infection of an animal infection of an animal with a virus that originated in a human. So basically, reverse is just the opposite of that. Rather than a virus starting out in a bird, for example, and then going to a human, it would be a virus that started out in a human and then can infect a bird, for example, or some other animal.